Welcome to Tomorrow Unveiled. Today we are opening up the research files for what we've dubbed the Immortality Update. If you have been following the longevity space, you know that the last few years have been remarkable, but 2025 and 2026, they've marked a truly seismic shift. The science is moving so decisively now, accelerating research from the purely theoretical into, well, to almost clinical reality. It's demanding that we revisit our entire understanding of what's possible.
It's an inflection point, certainly. The field of geroscience, which means, targeting the fundamental biology of aging itself rather than just treating the diseases that come from it, is gaining this critical momentum. Our shared mission today is to manage the fire hose of information. We're going to distill the massive amount of data our dedicated research team has compiled and really focus on three major interconnected frontiers. These are the ones that promise to extend not just how long we live, but crucially our health span, that period of life lived in robust, disease-free health.
And that's the core difference here, right? We're not talking about simply adding frail, unhealthy years. We're talking about engineering biological resilience. These three pillars, they represent a comprehensive approach to anti-aging, addressing completely different yet connected biological failures.
Exactly. The three revolutionary frontiers we've identified based on the cross-corroborated sources are, first, the really ambitious goal of reversing the clock via partial cellular reprogramming, an actual epigenetic reset.
A reset, I like that.
Second, clearing the clutter. This is the battle against senescent zombie cells and the chronic systemic inflammation they create.
Right.
And third, the new threats, which is a highly mechanistic frontier involving recently characterized novel forms of cell death that are linked to, of all things, metal dysregulation in our cells.
And crucially, the common thread connecting all these advances, from designing better rejuvenating molecules to optimizing personalized treatment plans, is the accelerating foundational role of artificial intelligence. AI is no longer just a helper, is it? It's the critical infrastructure that's turbocharging every single aspect of discovery and clinical translation.
It's the constant in the equation of accelerated progress. I mean, we are moving from observing aging to actively controlling its mechanisms.
Okay, let's unpack this and jump right into the deepest end of the pool, the attempt to reverse biological time. What exactly happens when a cell gets old on a molecular level, and how do researchers even begin to rewind that whole process?
Well, when a cell ages, its physical structure, its DNA, that stays the same, but its instructions, its identity, that suffers gradual decay. This is the epigenetic problem of aging. Think of your DNA as the hardware, right? The hard drive. Epigenetics is the software that tells the cell which programs to run, which genes to turn on, and which to keep quiet. Over time, that software it gets corrupted. It accumulates junk code, and the cell forgets how to be efficient and functional.
So the cell still has the blueprint for youth, but it just can't access the file. It's running old, buggy software.
Precisely. And this is where the research team highlights a particularly worrisome common aging pattern they've seen across multiple tissues. It's a pattern they've termed the mesenchymal drift.
Mesenchymal drift, that sounds ominous. Tell us more about what that functional shift means.
Functionally, it means cells across multiple vital tissues, the kidney, the liver, the lungs, they shift away from an epithelial state, which is, highly orderly, organized, and functional.
Like the carefully placed tiles on a floor. The pristine lining of an organ.
That's a perfect way to put it. They're dedicated to specific functions like filtering or absorbing. The mesenchymal state, however, that's much stiffer. It's more disorganized and inherently scar-like.
So instead of a perfectly filtering kidney lining, you get a patch of biological cement that doesn't do its job properly.
That's a great analogy. Scar tissue, which is largely mesenchymal in nature, is non-functional and stiff. This pathological drift, this shift from orderly functionality to scar-like stiffness, is strongly and repeatedly associated with the onset of chronic age-related diseases throughout the body. We're talking fibrosis, COPD, general organ failure. The key insight here is that this is a pattern of aging across tissues, which suggests a common underlying mechanism that we can actually target.
Which brings us to the core of the solution, partial cellular reprogramming. How do you safely fight this fundamental systemic drift?
The core mechanism involves introducing specific protein reagents, the most famous set being the Yamanaka factors. The goal is profound, but subtle, to safely turn back the clock by partially resetting the epigenetic landscape. The crucial word here is partial. You are trying to erase the age-related corruption, the accumulated epigenetic junk, without causing the cell to completely lose its original specialized identity.
Because if you fully reset the cell, you revert it to a state of pluripotency, essentially an embryonic stem cell, which is highly dangerous.
Exactly. Full reprogramming leads to a loss of identity and a high risk of teratoma formation, which is essentially an aggressive, disorganized tumor. The breakthrough here is finding that precise sweet spot, the shortest time or the minimum cocktail of factors necessary to restore youthful function without triggering that dangerous loss of identity. You want an old kidney cell to function like a young kidney cell, not turn into a universal, potentially cancerous blob.
And did the research team find evidence that this subtle partial reversal actually worked in a complex living system, like in an animal?
They found compelling, corroborated evidence. One of the most significant preclinical studies involved old mice that received periodic low-dose treatment using these factors over 7 months. And the results were not subtle. Tissues like the kidney and liver showed significantly lower expression of those destructive mesenchymal drift genes we just discussed.
So the cells were literally de-scarring themselves.
In effect, yes. This suggests that age-related decline wasn't merely halted or slowed down, which is the aim of so many traditional interventions. It was measurably reversed on a biological molecular level. This is a critical distinction for geroscience. It shifts the paradigm from maintenance to restoration.
The shift from finding these factors naturally to having AI design them, that fundamentally changes the economics of discovery, doesn't it?
Yeah.
Here's where the AI element becomes completely inseparable from the science.
It changes everything. This technology isn't staying confined to academic labs, it's being rapidly commercialized. Our research team highlighted specific biotech efforts, such as youth biotherapeutics, which is focused on developing a targeted gene therapy, YB002. They aim to use these partial reprogramming factors specifically to treat Alzheimer's disease by restoring youthful gene expression in the aged brain. The FDA has already supported their investigational plans to move toward first-in-human trials, marking a transition from theory to clinical reality.
But the real acceleration comes from optimization.
Yep.
I mean, it's one thing to use the factors nature gave us, it's another to design better ones from scratch.
That's the AI turbocharge. The research team noted crucial collaborations between established technology firms and longevity companies, specifically naming Retro Biosciences. These groups didn't just use standard AI, they retrained scaled-down versions of massive language models, like a variation of GPT-4o, for highly specialized protein engineering tasks.
How does a language model, which is typically used for generating text or code, become an expert molecular architect?
It's a fascinating pivot. They treat the protein sequence, the chain of amino acids, like a language. A standard AI language model learns grammar and context to predict the next word. When you retrain it on protein data, it learns the grammar of protein folding and function. It can predict which amino acid substitutions, the words, will result in superior structural stability and binding activity. This allows the AI to suggest millions of modifications computationally before a single one is ever tested in a lab.
So the AI is designing the instruction manual for the cell.
Exactly. It's designing superior tools for the reprogramming process. These AI-optimized factors, dubbed RetroPICX and RetroKLF variants, reportedly showed significantly superior activity compared to the original factors. This optimization accelerated the appearance of pluripotency markers days earlier than the standard methods. In a field where you often spend weeks waiting for biological processes, achieving key cellular markers days sooner is just an enormous efficiency gain.
And beyond sheer speed, what were the indicators that these AI-designed molecules actually made the cells healthier?
The most profound indicator was the improved performance in basic repair functions. The AI-optimized cocktail demonstrated significantly improved DNA damage repair capability in the treated cells. DNA damage accumulation is one of the foundational hallmarks of aging. So, by improving the cell's ability to fix its own damage, you are addressing the core problem. This kind of computational acceleration dramatically compresses the timeline required to move from basic discovery to a viable, optimized therapeutic candidate. It's setting a blistering pace for the field.
It sounds like we are moving from passively observing aging to actively designing our way out of it. Okay, so if we can reset the genetic instructions, that solves half the problem. But what about the cells that are just too damaged and refuse to die? That leads us straight into the conversation around cellular clutter, the zombie cells, and the second major pillar of longevity science.
That's the perfect segue because the research team's analysis shows unanimous consensus that cellular senescence is a fundamental core driver of aging. These are the cells that stop dividing, which again is good, but they refuse to die, which is very, very bad. They persist in tissues, hence that zombie cell nickname, creating massive systemic problems.
And the reason they are so damaging isn't just that they occupy space, but that they are actively toxic to their neighbors.
Precisely. They are chemical terrorists. They secrete a harmful, toxic cocktail of pro-inflammatory factors, tissue-degrading proteases, and dysfunctional growth factors. This is known collectively as the senescence-associated secretory phenotype, or SASP. This constant, localized chemical warfare spills out and creates chronic, low-grade systemic inflammation throughout the body.
And that low-grade fire has its own name, inflammaging.
Yes. Inflammaging is the smoldering engine that drives most of the chronic conditions we associate with getting older, whether it's neurodegeneration, the progression of many cancers, cardiovascular disease, or the accumulation of fibrosis. If you can control or eliminate these zombie cells and their toxic SASP secretions, you effectively extinguish the underlying fire that fuels chronic disease progression.
This sounds like a problem that requires a multi-pronged approach, which brings us to the dual therapeutic strategy the research team identified, the eliminators and the quieters.
The eliminators are the senolytics. These are drugs, such as the widely studied combination of dasatinib and quercetin, designed to selectively kill senescent cells. They target and exploit the specific anti-apoptotic survival pathways these zombie cells rely on, their biological Achilles heel. By selectively triggering cell death only in these dysfunctional cells, preclinical studies show a cleanup effect that dramatically enhances tissue function and extends healthy lifespan in animal models. It improves symptoms across dozens of age-related conditions.
And then you have the quieter approach, the Senomorphics. Why would you choose to quiet a zombie cell rather than just kill it?
Senomorphics, compounds like the common diabetes drug metformin or the immune suppressant rapamycin, don't kill the senescent cells, but rather suppress their harmful cyclin-A secretions. They effectively silence the inflammation they cause. The reason you might choose this is subtle but really important. Senescent cells sometimes play beneficial temporary roles, like in rapid wound healing or even regulating early tumor suppression. Quieting them rather than eliminating them entirely preserves those beneficial acute functions while reducing the chronic toxic spillover.
And the data suggests that combining these two strategies is the superior path forward.
It does. Combination therapies, Senolytics plus Senomorphics, are showing superior results because they offer both elimination and immediate inflammation control. Researchers are now exploring highly sophisticated sequential dosing protocols. The emerging best practice involves administering Senomorphics first. This suppresses the acute SASP factors and reduces inflammation before the main attack. Then Senolytics are administered to clear the cells.
Why the sequence? Why not just clear them all at once?
Because if you clear too many senescent cells simultaneously, they can release a massive short-term inflammatory burst as they die, a significant inflammatory rebound. By using the Senomorphics first, you reduce the inflammatory burden that these dying cells release, which makes the whole cleanup process safer, smoother, and it minimizes the risk of adverse reactions in the patient. That's extremely sophisticated, moving beyond a single drug to a choreographed therapeutic strategy.
That choreography points to the future of personalized medicine. This cellular clearance has also moved into the realm of advanced immunology, similar to how we treat some cancers.
Yes, drawing inspiration directly from cancer immunotherapy, specifically CRT cell therapy. Researchers have engineered specialized immune cells, we're talking about anti-UPAR CAR T cells, to act like precision-guided missiles. The UPR protein is a specific marker found on the surface of senescent cells. By training T cells to recognize and attack this marker, they can seek out and eliminate senescent cells with incredible specificity, at least in mice.
So we're essentially turning the body's own immune system, which usually weakens with age, into an incredibly precise, personalized cleanup crew for aging. What specific benefits did this immunotherapy approach show?
Beyond just general clearance, it showed deep functional benefits, particularly in the gut. They thought great improvement in the overall metabolism of the mice, which suggests systemic benefits, but notably it stimulated cell growth and repair in the aging intestine. This led to reduced inflammation and vastly improved nutrient absorption, a major factor in maintaining health as we age. It points toward using the immune system not just to fight disease, but to actively regenerate tissue health.
And speaking of dramatic rejuvenation across multiple systems, the findings from that large-scale macaque study, highlighted by our research team, that has to be a huge validation of the entire field.
This truly was a landmark moment for geroscience. Chinese scientists used genetically engineered senescence-resistant stem cells, or SRCs, in aged macaques. The key engineering step was to modify these stem cells to produce a specific protective protein. This protein enhances the transcriptional activity of protective genes, which lets the SRCs withstand the harsh inflammatory microenvironment of aged tissue, a critical failure point for natural stem cells.
I want to spend a moment on the scope of this. This wasn't a localized treatment. What was the impact across the entire organism?
The effects were dramatic and body-wide. The SRC treatment provided enhanced regenerative capacity, essentially rejuvenating the stem cell niche. They conducted exhaustive analysis and observed that over 50% of the 61 tissues they examined showed signs of reversal or rejuvenation. This wasn't just skin or muscle. It included deep systemic effects.
Give us some of the most surprising examples of rejuvenation outside of just generalized metabolism.
The effects on the central nervous system were highly compelling. The treatment mitigated age-related brain shrinkage, particularly in the hippocampus, the brain's crucial memory consolidation center, and restored brain connectivity to levels comparable to younger animals. Furthermore, the regenerative capacity extended to bone health, reversing age-related bone loss, such as in the alveolar bone structure around the teeth. This suggests profound implications for physical resilience and quality of life. They also noted significant reductions in systemic inflammation markers and DNA damage across the board.
That is the ultimate validation of the thesis. If you can improve the quality and resilience of your foundational stem cell pool, the benefits don't just localize, they ripple outward, rejuvenating the entire organism.
It connects back perfectly to the core challenge of aging: stem cell exhaustion. Regular stem cells have some anti-aging effects, but they often fail when faced with the chronic stressors of an aged, SSP-driven inflammatory environment. The engineered SRCs, however, appear to be bulletproofed against that harshness, offering a superior and sustained regenerative capacity.
Beyond the systemic clearance, we also have data on targeting specific inflammatory drivers linked to senescence, which leads us to the investigational therapy involving interleukin-11, or IL-11.
The anti-IL-11 monoclonal antibody represents a targeted precision approach. Interleukin-11 is a key cytokine, a signaling protein that has been strongly linked to aging-associated diseases, particularly fibrosis. Fibrosis, remember, is essentially pathological scarring, that scar-like mesenchymal drift we discussed earlier. It causes organs to stiffen and fail.
So if you block IL-11, you interrupt the signaling pathway that tells the body to create scar tissue and stiffness.
That's the hypothesis, and the preclinical data supports it. The therapy demonstrates robust anti-fibrotic activity across multiple disease models, including conditions like pulmonary fibrosis and hypertrophic scarring. Crucially, the research team found that modulating the IL-11 pathway has broad relevance in conditions linked to cellular senescence, frailty, and overall longevity, because fibrosis is such a ubiquitous consequence of chronic aging.
And this isn't just theory anymore, it's moving through the pipeline.
Correct. The therapy is now in phase 2 clinical trials for pathological scarring. If successful, this therapy offers a potential systemic treatment for age-related stiffness and dysfunction, further validating the strategy of targeting specific fundamental aging mechanisms like inflammaging and the resulting tissue stiffening.
We've covered the fundamental epigenetic reset and the incredibly sophisticated cellular cleanup operation. Now we need to move to the third frontier, which is perhaps the most highly mechanistic and chemically focused. The collapse of metabolic stability driven by metals, which gives rise to fundamentally new forms of cell death.
If we connect this to the bigger picture, aging isn't just about accumulated physical damage or zombie cells. It's fundamentally tied to metabolic collapse, especially mitochondrial dysfunction and chronic oxidative stress. These processes are deeply intertwined with how the body handles transition metals, metals like iron and copper, which act as critical cofactors but can become toxic when they get dysregulated.
Which brings us to two pathways that sound like they belong in a chemistry textbook, but are now central to geroscience: Feroptosis and Cuproptosis.
Indeed. Let's start with Feroptosis. Defined over a decade ago, this is an ion-dependent form of regulated cell death. It's not apoptosis, which is neat and tidy cell suicide. Feroptosis is messy. It's driven by excessive lipid peroxidation, that is, the polyunsaturated fats in the cell membrane go catastrophically rancid due to the failure of the cell's primary antioxidant system, the GPX4-GSH axis.
Wait, I want to clarify that mechanism, GPX4 and GSH. What are they and why are they so central to preventing this cellular rancidity?
So GSH, or glutathione, is the cell's master antioxidant molecule. GPX4, glutathione peroxidase 4, is the enzyme that uses GSH to neutralize highly damaging lipid peroxides, those rancid fats, before they can destroy the cell membrane. If you run out of GSH, or if GPX4 is inhibited, iron is free to catalyze a runaway reaction, accelerating the lipid peroxidation until the cell membrane just bursts. It is essentially an uncontrolled rusting of the cell catalyzed by free iron.
That's a very clear pathway for targeted intervention. Now let's talk about the newer, even more complex pathway, cuproptosis.
Cuproptosis is copper-dependent, and it was only characterized in 2022, marking a major new piece of the aging puzzle. While Feroptosis is about lipid damage, cuproptosis is triggered by copper overload, disrupting specific mitochondrial metabolic processes, primarily something called protein lipoylation.
Protein lipoylation, that sounds incredibly niche.
It is, but it's vital. Lipoylation is the process of attaching a fatty acid, lipoic acid, to key enzymes within the mitochondria. These enzymes are essential for generating cellular energy through the TCA cycle. When excess copper binds to these lipoylated proteins, it causes them to aggregate and clump together. This leads to massive, fatal stress within the cell called proteotoxic stress, specifically within the mitochondria. It causes total energy collapse and cell death.
So we have two different metal-driven train crashes. Iron causes the membrane to rust and fail, and copper causes the mitochondrial engine to seize up with clumped proteins. This raises an important question. Why should you care about iron and copper dysregulation specifically in the context of aging?
You should care because these two pathways are not separate biological silos. They are interconnected axes within the broader metal redox metabolic network. Aging drives a fundamental dysregulation of both iron and copper homeostasis, meaning the cell loses the ability to properly store and manage these elements. This leads to the accumulation of labile or redox active metal pools, metals that are free to catalyze harmful free radical reactions.
So the older cell isn't just inefficient, it's sitting on a powder keg of mismanaged metals, just waiting for a trigger. How does senescence, the zombie cells we just discussed, link back to this metal problem?
The links are profound. For example, senescent cells accumulate high levels of iron because a cellular self-eating process called ferritinophagy, which clears stored iron, becomes impaired. This accumulation expands the labile iron pool within the senescent cell, promoting phenton chemistry and lipid peroxidation, which significantly lowers the threshold for feroptosis, making those cells fragile.
So the zombie cells are not only toxic from their SEC secretions, but they're also inherently vulnerable to death by rusting.
Exactly. And the copper pathway connects too. Copper dysregulation has been shown to destabilize GPX4, the primary enzyme that protects against feroptosis, making the cell vulnerable to both mechanisms of death simultaneously. It's a vicious cycle of metabolic breakdown.
That suggests that if you treat the metal dysregulation, you can interrupt the cell death signal before it even starts, which is the definition of preventative geroscience.
That is the powerful therapeutic premise. Potential interventions involve pharmacological modulation, using specific molecules, known as chelators, to bind excess metals. For iron, we can use iron chelators to pull the free iron out of the labile pool. For copper, drugs like tetrathiamolybdate can reduce systemic copper levels, protecting against tissue damage and cuproptosis.
And what about existing accessible interventions that intersect with these vital pathways?
A critical, widely available strategy involves boosting glutathione, or GSH. Since GSH is the principal defense against feroptosis, it's the fuel for GPX4 replenishing it can dramatically improve cellular defense. Our research highlights the use of N-acetylcysteine, or NSC, and similar cysteine donors, which serve as direct precursors to replenish GSH reserves. Preclinical evidence strongly supports its use in models of neurodegeneration, demonstrating a clear link between bolstering this axis and protecting against oxidative and metal-driven injury.
This level of detail in understanding cell death is clearly moving us toward precision medicine. How are researchers translating this knowledge into identifying who is most susceptible?
It's the next logical step. The research team stressed the emergence of biomarkers for precision medicine strategies. Clinicians can now look at specific indicators, measuring serum ferritin, which reflects iron storage, analyzing copper to zinc ratios, which are indicators of overall metal balance, and measuring GPX4 activity. Identifying individuals who are accumulating labile metals or whose antioxidant systems are compromised allows for targeted interventions to mitigate metal-driven cell death before it manifests as neurodegeneration or organ failure. This is the shift from reactive medicine to predictive health maintenance.
We've covered a lot of incredibly dense material here on Tomorrow Unveiled, from resetting DNA to clearing damaged cells and stabilizing cellular energy systems. If you're finding this insightful, please take a moment to like this video, subscribe to Tomorrow Unveiled, and consider hitting that new hype button to help us reach more viewers. And of course, be sure to turn on notifications so you never miss an update on the future. We'd established that the fundamental biology of aging is cracking open. Now let's switch gears and talk about the infrastructure that makes this revolution possible, which brings us back to our great accelerator, artificial intelligence, and how it's changing the face of diagnostics and drug design. What's fascinating here is the speed at which AI is moving from being a novelty to being critical infrastructure. We already talked about it designing better partial reprogramming factors, but it's extending into the core drug discovery process itself. How does AI identify a new drug target faster than a team of chemists?
Companies like Gero are using what they call physics-informed AI models. Unlike traditional machine learning that just looks for correlations in vast data sets, these models are trained on human data sets, but are constrained by known biological laws, the physics of the cell, if you will. This combination allows them to identify new therapeutic targets aimed specifically at the root causes of aging with much higher fidelity and a significantly reduced false positive rate.
That sounds like a radical shortening of the discovery pipeline and a huge boost in confidence for pharmaceutical companies.
It is. The research team noted that this has led to significant joint research and licensing agreements between these AI-driven companies and major pharmaceutical partners. In one case, an agreement allows a pharma company to create novel antibody drugs based on targets discovered by the AI platform, effectively outsourcing the hardest part of the discovery phase. This dramatically reduces the decades-long timeline and high failure rate that traditionally plague drug development.
Beyond discovery, AI is also transforming diagnostics, particularly in the realm of oncology, by leveraging massive databases of aging-related factors.
Absolutely. Machine learning models are analyzing large, publicly available data sets, such as the Cancer Genome Atlas, to identify prognostic signatures based on aging-related factors. For example, AI models can analyze the expression patterns of reactive oxygen species, ROS-related genes, those same oxidative stress factors we mentioned earlier, to predict the prognosis of bladder cancer patients. Similarly, they can calculate cellular senescence scores within a tumor to predict outcomes in lung adenocarcinoma.
So aging biomarkers are now becoming diagnostic tools for unrelated diseases.
Precisely. This shift provides clinicians with predictive tools they simply didn't have a few years ago, allowing them to tailor treatment plans with greater foresight based on the patient's underlying biological age and vulnerability, not just their chronological age.
This personalization is taken one step further with digital twins. Explain the concept of a healthcare digital twin and how it translates to real-world care.
The healthcare digital twin is a truly transformative concept, a dynamic, real-time virtual model of an individual patient. This model integrates everything: your genetic blueprint, continuous inputs from wearables, electronic health records, proteomics, even metabolomics. It's a constantly updated, living, breathing virtual replica of your current biological state and future trajectory.
And the application here moves care from reactive guesswork to predictive personalized simulation.
Exactly. These models enable clinicians to run virtual trials. Instead of administering a drug and waiting to see if it works, a doctor can simulate that process on the digital twin. For instance, they can simulate how a patient's heart might respond to specific drugs tailored to that individual's unique biology, identifying potential side effects or optimizing dosage before ever administering the medication. This minimizes risk and maximizes efficacy.
That dramatically shifts the odds in the patient's favor. The technological advancements aren't stopping at personalized modeling. They're also being deployed to rejuvenate one of the most vital and most rapidly declining systems in the aging body, the immune system.
That is the story of immunosenescence, the profound weakening of the immune system with age. This makes older people less responsive to vaccines, highly vulnerable to novel diseases like COVID-19, and less capable of fighting cancer naturally. Researchers are now deploying new mRNA technology, the same platform used for the highly successful vaccines, to address this decline.
How does this mRNA approach manage to reverse the decline of the immune system?
The decline is largely due to the shrinking and dysfunction of the thymus, the organ responsible for maturing T cells, the critical soldiers of the immune system. Researchers used mRNA to temporarily transform the liver in mice into an ectopic factory, a temporary manufacturing site, for producing crucial immune regulatory factors that normally come from the thymus.
So they use the liver, which is highly robust even in old age, to fill the production gap left by the failing thymus.
Precisely. By essentially creating a temporary secondary source of these crucial factors, they were able to restore the formation and maturation of new T cells. It's a really elegant bypass solution for an aging organ.
And what were the real-world results of this rejuvenation?
The results were highly encouraging. The older animals that received this treatment developed robust, youthful immune responses. Critically, they responded far better to common vaccines, showing higher antibody production. And most importantly, they showed much higher survival rates when treated with cancer immunotherapy drugs, specifically checkpoint inhibitors, compared to mice that only received the cancer drug.
That suggests that aging itself is the limiting factor in many of our best cancer treatments. And if we can rejuvenate the immune system, we can dramatically improve outcomes for older patients across the board.
It indicates that immunosenescence is targetable and reversible. The mRNA technology offers a fast, temporary, and highly programmable way to bolster the resilience of older patients against multiple threats, from infectious disease to cancer. The science is clearly racing ahead with incredible speed, as we've detailed through 3 distinct frontiers. But there's a serious, systemic roadblock we must address. The regulatory environment is struggling to keep pace with these revolutionary advances.
That raises a critical question. If these treatments can reverse aging in animal models and are moving into phase 12 trials, why aren't they moving into comprehensive human trials faster? What is the core challenge with regulation?
The fundamental issue is that current regulatory agencies, like the FDA and the EMA, they're structured to approve therapies for single, defined diseases, specific cancer, type 2 diabetes, or macular degeneration. They do not classify aging as a disease itself. This lack of formal recognition prevents the field from designing standardized systemic clinical trials.
That makes designing endpoints incredibly difficult. The FDA needs a clear, measurable outcome for approval. If you're treating aging itself, what do you even measure?
It creates a massive barrier for gerotherapeutics. How do you prove efficacy? You can't run a 50-year trial. Researchers are proposing workarounds. The TAME trial, which investigates metformin for multiple age-related diseases, set a crucial precedent by targeting the mechanisms of aging, rather than a single disease endpoint. But a comprehensive, harmonized framework for systemic aging is still missing globally.
So what pathways are being proposed by the research team to bridge this regulatory gap?
They suggest 2 key solutions. First, formally recognizing the mechanisms of aging like senescence, chronic inflammation, and epigenetic instability as legitimate targets for intervention. This allows the trial to focus on biological reversal, not just disease prevention. Second, developing regulatory frameworks that can accommodate systemic aging biology and establishing clinical endpoints that reflect health span.
What does a health span endpoint look like in practice?
It moves away from waiting for a catastrophic event like a heart attack. Instead, it measures metrics like intrinsic capacity or resilience. Intrinsic capacity might include measures of muscle strength, cognitive speed, walking velocity, lung function. If a therapy improves or preserves the patient's intrinsic capacity, their ability to function robustly, that should be a recognized, measurable outcome for approval. It reflects true health span extension.
If these therapies become a reality, and based on the data they're rapidly approaching, what does this all mean for society? The ethical and societal questions are just enormous and potentially destabilizing.
The research team highlights the most critical ethical concern raised across all sources: access inequality. The high cost of developing these cutting-edge therapies, gene therapies, CRT cells, AI-optimized drugs, risks limiting them exclusively to the wealthy. If only the top 1% can afford to access robust systemic health extension, it risks dramatically widening socioeconomic divides in health outcomes.
We could face a future where longevity becomes the ultimate status symbol, and the rich can afford to outlive the rest by decades.
That's the grave risk, and the resulting systemic strain would be daunting. Extended healthy lifespans will put enormous pressure on current retirement systems and pension funds, which are fundamentally designed around mortality assumptions from the mid-20th century. Healthcare infrastructure, currently designed for acute care, would need a complete proactive overhaul. And the job market faces potential disruption as older workers remain active and productive for decades longer, potentially blocking career progression for younger generations. These are risks that must be proactively addressed through policy now, not later.
That brings us to a crucial point of balance, offering a necessary counterbalance to the intense focus on high-tech drugs, the power of non-technological intervention.
It's vital to remember that biology always responds to environment and lifestyle. Our research team provided a compelling anecdote from a longevity expert, Dr. David Furman. He found his biological age, specifically his inflammatory age, measured by systemic biomarkers, was chronologically older than he was. He was 39 chronologically, but his inflammatory age was 42, reflecting high stress and poor health habits.
And he took some pretty radical steps to address this biological debt.
He did. He quit his high-stress urban life and moved his family to a remote forest cabin for 3 years. He embraced a strict restorative lifestyle, organic, whole food eating, intensive stress reduction, exposure to nature, and, notably, a rigorous digital detox. He used only candlelight in the evenings to restore his circadian rhythm.
And the change was truly dramatic.
Chronologically 39, with a biological age of 42, he reversed his biological age by a decade, down to 32, in just 3 years. He eliminated chronic migraines, saw a surge in focus and productivity, and significantly improved those systemic inflammatory biomarkers. It underscores that we already possess powerful, non-proprietary tools for health span extension.
That's phenomenal. It demonstrates that while technology aims to fix the cellular problems of aging through drugs, the environmental and lifestyle context is still the primary lever for most people.
Absolutely. For the vast majority of people globally, longevity depends not on expensive proprietary gene therapies, but on collective responsibility and public policy. It's about ensuring access to clean air, infrastructure that supports active cities and daily movement, affordable, nutritious food access, and effective stress management systems. The public health perspective frames longevity as a foundational social achievement, not merely an individual optimization task achievable only with a massive bank account.
This has been an incredibly dense and comprehensive update on the frontiers of longevity science. Let's quickly summarize the key takeaways that you, our listener, need to carry forward from all this.
First, partial cellular reprogramming is moving rapidly closer to human trials. It's being accelerated by AI optimization models that are designing superior, more efficient biological tools, offering the potential to reverse age-related decline at the epigenetic level.
Second, the chronic issue of zombie cells, cellular senescence and inflammaging, is being tackled with sophisticated dual strategies: Senolytics for clearance, Senomorphics for silencing, and advanced immune approaches like engineered CRT cells, showing dramatic results in animal models across multiple organ systems.
Third, new targetable frontiers involving fundamental metabolic aging pathways, like the metal dysregulation driving Feroptosis, lipid rancidity, and Cuproptosis, mitochondrial protein collapse, are being addressed with specific chelators and supplements like NEC to stabilize the cell's internal environment and boost its natural defense systems.
Finally, while AI and digital twins are turbocharging the discovery and personalization of these therapies, the primary challenge remains translating these advances equitably. We have the regulatory bottleneck that fails to recognize aging as a target, and the critical risk of creating a massive longevity gap between the wealthy and the common person.
And that leads to our final provocative thought for you to consider as you digest the potential of this technology. The greatest longevity breakthrough might not be the technology that lets the rich live to 150, but the political will to treat aging as a fundamental public health challenge, ensuring every person has access to the conditions, the environment, the nutrition, the stress reduction that make a healthy life to 100 possible. That political will, that is the ultimate gerotherapeutic.
We love hearing your thoughts. Whether you're on YouTube or your favorite podcast app, leave a comment and let us know what you think about extending health span and the challenges of equitable access to these new therapies. And for our audio listeners, if you're listening on a podcast service like Apple Podcasts or Spotify, we'd be incredibly grateful if you could leave us a rating and review for Tomorrow Unveiled. It truly helps us reach new listeners. Thank you for diving deep with us into Tomorrow Unveiled. We'll be back next time to explore the next frontier.